I'm going to talk about the opposite of rare animals in small populations that are hard to keep track of and hard to get samples from. So Sean asked me to talk in general about sort of lessons from genetic conservation in trees. I'm going to interpret that fairly liberally, and I'm going to talk about a number of things. First of all, I'm going to talk about a little bit about neutral versus adaptive patterns of variation, and for the kinds of species I work with, how useful these types of information are for meeting the challenges of managing and conserving species. I'm going to talk about how we get to genomic signatures of adaptation, whether we can get good information from genomic data on patterns of adaptation. I'm going to talk about how we do things, how we have done things in BC in terms of assessing in situ conservation, where we've been using the opposite of very expensive genomic tools. We've been using existing climatic and botanical plot data. And I'm going to talk about climate change. I'm talking about trees. They live a long time. They're stuck in one place. And long-lived trees are experiencing more climate change, a faster rate of climate change than anything else on Earth, because it's the rate of environmental change you can withstand has to do with the amount of change in a generation. And these are long-lived things. So climate change is pretty much overwhelming a lot of other concerns and the associated changes in pests and pathogens we're getting. Okay, so there's the very odd case where we've got extremely restricted distributions. We have some species that have a few fragmented populations, but for temperate and boreal species that I deal with, we are mostly dealing with very large continuous populations. So simply defining populations on any kind of demographic basis is very difficult. But we see, as an example of a species like this, with this kind of structure, we have something like Chihuahua spruce in Mexico. And here we are, a bunch of people interested in forest conservation genetics, visiting this species in Durango, Mexico. And here we have the one zoom image. I'm going to talk a lot about these guys today, which are far from endangered. But here we have this little leaf on this spruce branch that is Picea chihuahuensis. And we know from isozyme work done back in the 80s, big FST among populations. There are few populations. There are few trees in these populations. And we don't really need much more genetic data to know we've got a problem here that needs a lot of attention. And certainly the people in Mexico are working on ex situ collections and protecting additional stands, etc. But for most of our widespread species, we've got strong patterns of local adaptation. These are trees from different populations grown in a common garden, and they show very strong adaptation to climate. And that's what we see over and over again in our widespread conifers. We see strong adaptation to broad climatic gradients, particularly to low temperatures. So, neutral markers, FST. Here we have a bunch of widespread tree species. Here are the FST values from a variety of markers for these species. And these are QST estimates for a variety of traits. For most of our species, for the traits involved in local adaptation, we have much, much higher QSTs than FSTs, to the point where really FST for these species doesn't tell us much about what we need our management units to be. And this has always been our problem with markers. If we see a big FST difference, then we can interpret that, but if we don't see one, it doesn't mean we don't need to manage things separately. Here's an example, Lodgepole Pine. Okay, I'm going to show you a lot of Lodgepole Pine and what I call interior spruce. These are economically and ecologically important species. Therefore, we got the money to do a big genomic study on them. These are not in any way at-risk species, but here we have, so we did targeted sequence capture for about 23,000 genes in over 250 populations of 2 different species that I'll be talking about. One of them is Lodgepole Pine. We had a set of putatively neutral SNPs here that we used to look at population structure, and we have an extremely low FST for those SNPs. This is what the spatial pattern for the first dimension of the multi-dimensional scaling looks like. We've got a bit of an east-west gradient here. We do have a different subspecies on the coast, but that's the pattern. When we look at phenotypic data, here we have artificial cold hardiness test data, and the blue circles say things were very cold hardy, and the red say that they were not cold hardy, and we have a general pattern that agrees strongly with modeled climate surface for extreme minimum temperature. Makes sense. Let's look at another, the other species is actually a species complex that we looked at. Here we have stronger hybrid structure. Oh, the FST is not on there, but it's about 0.06 across these SNPs, but we've got 2 species and they're hybrids. White spruce in blue, Engelmann spruce in red, and then hybrids. And we've got this generally northeast to southwest pattern. This is British Columbia, this is Alberta. This area is about as big as California, Oregon, and Washington put together. So it's a big geographic area we're talking about here. So we get this general pattern. That's the population structure. Again, for MDS, this is what the phenotypic traits show us. Same pattern. So what do we usually do when we're looking for signatures of adaptation in genomic data? We adjust for population structure. What's going to happen if we adjust for this population structure? We will find nothing. So let's look at what that looks like. So just to walk through how we've looked at this problem, first of all, we had 23,000 genes. We wanted to find ones involved in local adaptation. The first thing we did is we identified what we call we took SNPs. We had about 1 million SNPs. We looked at genotype environment associations. We looked at genotype phenotype associations. And we found the strongest correlations. And then we looked for the genes that had more outlier SNPs per gene relative to the number of SNPs in the gene. So not just looking for single SNPs, but looking for multiple SNPs in genes that were good outliers. And this reduces our number of things we're looking at down to genes from SNPs. So this is the Lodgepole Pine. This is genotype environment interactions for 22 different environmental variables. And the purple bars are genes that we found when we corrected or didn't correct for population structure. The blue are non-corrected. The red are structure corrected. You see we picked up fewer things in the structure corrected category, but we still had a fair representation of genes here because we had pretty weak population structure in the Lodgepole Pine. When we do that for the spruce, this is what it looks like. We lose everything. Well, we lose almost everything. These are non-corrected number of top candidate genes, many genes. We correct and we lose that signature. So this is a huge problem for using genomic data to detect signatures of adaptation. The way we got around it was we compared these 2 species. And I think that as people get more and more genomic data, they are going to be able to do this more and more. And comparative approaches let you sidestep the population structure problem entirely. So what did we do? Well, first of all, we said, okay, we've got, and we focused just on a small set of genotype environment associations and genotype phenotype associations, all associated with low temperature adaptation. And so we said, okay, we've got these top candidate genes for pine that we found using raw correlations, non-structure adjusted correlations. So you know there's a whole bunch of false positives in there, probably. We said, if we look at spruce, raw correlations, do we find these top genes? And then we looked at the spruce, raw correlations, and we turned around and looked at those genes in pine and said, do we find these in pine? We found 47 genes in common. Now, is that a lot or is that a little? I don't know. We found a few hundred genes in each species, but 47 seem like quite a few to us. It's quite a core. So how do we use that? How do we use those 47 genes to delineate populations, spatial units for conservation, to understand how these populations can adapt to climate change? I think that, for me, is the grand challenge of next-gen approaches for the things that I work on. So, do these give us the same patterns? Does the genomic data give us the same patterns of variation that we get from phenotypic data? Well, these are populations clustered into adaptive groups using a multiple regression tree approach, work done by Andreas Hammond and a method proposed by him. And in this regression tree, you end up with these groupings of populations. And we did this also for phenotypic traits. I won't show you the data. And the groupings were extremely similar. And the spatial distribution was very similar. And in fact, this spatial distribution is, at least in British Columbia, is not that dissimilar to the breeding zones for these trees that are based on phenotypic traits. So we seem to be getting similar signals. We're getting similar climate drivers, which I think is really important. The genomic data can tell us which environmental factors are important for adaptation. And that's something that I think is a big advantage. Okay, so for reforestation, seed, this is just for one species, is managed within these zones because we're maintaining locally adapted populations. We use a different framework I'll talk about in a moment for the genetic conservation, but this is a distribution of, well, current, this was actually 1961 to 1990 climate normals, and this is a middle-of-the-road projection for 2050. So this is our major problem, is that we have populations that are adapted to historic climate and not to new climates. Now, there's lots of reason to think they're going to adapt. Things with large population sizes, lots of variation, but it is going to take some time. Things that do not have large populations or are facing other challenges associated with climate, like pests and pathogens, are going to have a much harder time. So for the genomic data, this is what I think we need. Right now, we need to understand if we have allele frequencies within a focal population, how can we translate that into information about the capacity for that population to adapt to new conditions? So we need information on how fast climate is changing locally and how heterogeneous climate is locally. So the concept of velocity of climate change is very useful here. We can compare populations in different climates to understand the degree of mismatch between two populations. And if we have phenotypic data, we can just use the breeder's equation with heritability and say, how can you shift this to there? How do we develop an analog for that with multi-SNP data? This is what I would like to know. We need to incorporate population size, because whether populations can get through that evolutionary rescue is going to depend on how big they are. Because if they hit the bottom, it's a U-shaped trajectory. If they hit the bottom before they get out the other side, well, they're not going to get through. And we need to understand something about gene flow levels and how gene flow is redistributing adaptive variation. Just a shout out for this paper by Matt Fitzpatrick and Stephen Keller about some approaches to look at allele turnover over environmental gradients, and I think this is a way we could use genomic data like this, and we're exploring this method with our data. I probably shouldn't even talk about that. Other things we can get from genomic data. I know this was in the notes that Sean wrote up, but no one's mentioned it, but not all alleles are equal, and with things that have historically outcrossing populations, there's a lot of deleterious rare alleles in there. And we're discovering this for spruce and pine. About 12% of the alleles we've identified for the 23,000 genes that we sequenced in spruce actually have a deleterious signature when we put them through this Microbian sequence analysis approach, which I won't go into. Okay, so that's the BMW version, the genomic data that I was talking about. This is how we've actually characterized the adequacy of genetic conservation for BC's 50 or so tree species, depending on how you classify a tree. We've used an ecological framework. We haven't used genetic data. So we have an ecological classification system. It's hierarchical. It's got zones, subzones, variants. And we're looking in these zones, there's 16 zones, we're looking to see if there are 3 populations in protected areas for every species in each zone it occurs in. And that those populations have, ideally, we were shooting for 5,000 capacity habitat for 5,000 mature trees. You can't do that with most of the things that a lot of you are working on. But we've got large areas and large protected areas. So we roll up botanical plot data on not just occurrence, but coverage, percent area coverage of all the tree species. We integrate that with the ecological data, and we integrate that with the protected areas. And that's how we classify it. It's pretty simple. Is this good enough? This isn't next generation. This is old generation. And then we say, okay, what does that matrix look like? Where are the holes? But we've extrapolated representation across those ecological areas. And what we found is this works pretty well. This is the first cut at that. This works pretty well for things that grow on typical sites for these ecosystems, what we call zonal sites. Doesn't work well for habitat specialists, like riparian zones in very dry areas. We do a terrible job of estimating coverage because of the way the botanical data was collected. But we have this framework, ecological zones, species, for in situ and for ex situ. These are seed collections in the seed bank. And that's the framework that we've used. So we haven't used genomic data. We haven't even used population genetic data because the population genetic data tells us for most of these species that we have an FST of some rather low number. And we don't really want to plan around those FST numbers. Last point. I think this is my last point. Whitebark pine. I'm not going to show you any of the work we've done on whitebark pine, but whitebark pine is a widespread species, very important in the West, high elevation, anchors tree line in many areas. Grizzlies eat the seeds, which makes it a sexy tree, but a lot of other things are dependent on it too. And whitebark pine is our canary in a coal mine. So it's suffering from, number one, an invasive introduced pathogen, white pine blister rust. So populations are collapsing across the range. It has been listed as endangered in Canada, even though there are millions and millions of whitebark pine, but the populations are collapsing. Many of them, not all of them. Number two, beetles. Mountain pine beetle outbreak has hammered whitebark pine in a lot of areas, along with other pines. Number three, climate change. The other species are moving uphill and are getting more and more competitive in those environments. So it's this triple threat. And genetic data for resistance, there is about maybe average of 5% resistance to white pine blister rust in these populations. If we had markers for resistance, that would really help. Other types of genetic data, probably not so much, in fact. There's not much population differentiation. It's species with weak population structure. It even has relatively weak local adaptation. But that's the kind of challenge that we're facing, and it's a big one. I have one last example. Another problem. You may never have heard of Abies religiosa, Oyamel fir. But this is the fir, the true fir, that monarch butterflies overwinter on. Here's the monarch butterfly reserve. These are projections of the climatic niche of Oyamel fir. This is the historic distribution or contemporary. These are suitable habitats. And the contraction, so the yellow is what'll be suitable in year 2030 under the assumptions that Aquatimuk Saenz Romero made. Orange 2060 and red 2090. So by 2060, the monarch butterfly reserve is no longer within the climatic niche of Oyamel fir. So what are you going to do? So that's a climate change problem, but it becomes an issue of whether we're going to move trees. The reserve needs to move or expand. They're planting this species in new areas to try to create habitat, but it's going to take a long time to create the big trees that these monarchs overwinter on. And it's not just that the monarchs overwinter on the trees. They overwinter on the trees because those trees inhabit a climatic niche that allows the monarchs to get through the winter. The temperature and the moisture. So I guess that's another canary in the coal mine. Okay, so what I see as needs, this is what I do for fun. Well, no, that's not me climbing. We did some really fun sampling of trees this summer that were over 80 meters tall. That was the tops. That was really fun. You can ask me about that at the break. Okay, what we need methods for really incorporating adaptive genomic data in delineating conservation units, in understanding adaptation, in understanding potential for adaptation to climate change. We need to better understand if and when we can just use climatic or ecological data. And I think with our spatial, we just have to do things. Everything needs to be spatially explicit in the context of the things we work on. And I'm really glad that we've got spatial ecologists here who can help us with that, like Marie José. I didn't even talk about another area, which is tree species. So we've got tree species in breeding programs. Most of the things in breeding programs have been bred since long before we had tools to study the process of domestication. But some trees are in this very early stage of domestication, and we have a lot of work to do to understand what the effects of that domestication are. And finally, call to arms, rear edge populations, seed collections, ex situ collections, absolutely should be #1 priority. And I really commend Sean for the work he's done on ex situ criteria and or sampling strategies and pushing that, because that's one of these things like conservation genetic, population genetic parameters. Sort of we still use things from the 80s. The ex situ collection standards really need to change, and the priorities for us need to be driven by climate change. I know in the US we don't know if climate's changing, and we will know at about 10 o'clock tonight. And this is as close as I can get to a pantsuit, but yes, it's a political statement. So, but anyways, we really need to focus on those collections. In Mexico, the colleagues see the trees the southern margins populations aren't producing seed. They can't get seed from them anymore. That's a signal. So anyways, that's enough for me. Thank you very much.